All right, so we left off talking about transcription, and so I just wanted to kind of review the concepts that we discussed last Wednesday. Remember, when we're talking about transcription, we're talking about using DNA as a template in order to make RNA. And so there's going to be three phases that are going to be involved in transcription. There's an initiation stage, an elongation stage, and a termination stage. In initiation, the first thing that's going to happen is our enzyme that is in charge of transcribing. The name of that enzyme is RNA polymerase. And so RNA polymerase is going to polymerize the making of new RNA molecules. And so that RNA polymerase has to have a spot to bind onto the DNA before it can start turning that DNA into RNA. And so that spot at which that RNA polymerase binds is called the promoter. Remember, the promoter is going to be just ahead of the gene. That's just a regulatory region of DNA, one of these non-coding regions of DNA. And so its sole purpose is for the RNA polymerase to come and bind and set up shop before transcription begins. And so each gene has their own promoter. So for every single gene that's getting transcribed, that RNA polymerase will bind to that gene's promoter and then wait for the signal to go ahead and start transcribing. Now, once initiation is complete and you have the RNA polymerase on the promoter, the next stage is going to happen is elongation. And so in elongation, this is where the enzyme is actually going through using the DNA as a template and making the RNA molecule. So if you look at this DNA, this is the double helix in blue right here. The actual function of the enzyme is to look at the strand of DNA. So in this case, we're looking at CCGGAC. So what RNA polymerase is actually doing, it's reading this strand of DNA, and then it's making a strand of RNA based on the complementary base pairing. So if it sees a C in the DNA, it's going to put in a G in the RNA. Sees another C, it's going to put in a G. If it sees a G, it's going to put in a C, so forth and so on. So transcription is just looking at a string of DNA and then making an RNA molecule based on those complementary nucleotide pairings. And then once elongation is complete, the next phase is going to be termination. And so termination is going to signal the end of transcription. And so what's going to happen at the end of the gene, you're going to have another one of these non-coding regulatory sequences of DNA. In this case, it's called a terminator. So once the RNA polymerase finishes making the RNA molecule and it comes to the end of the gene, it'll run into this terminator region. And once that RNA polymerase comes in contact with the terminator region, it knows to stop transcription. And so at that point, the RNA polymerase will get up and leave, and then you're left with a nice shiny molecule of RNA. And so remember, we also talked about three things that needed to happen in order for us to process RNA. Remember, we have to protect it from all of those bad enzymes that are floating around in the cell that's capable of destroying it. And so we're going to protect both of the ends of the RNA molecule that we just made. So we're going to add a 5' cap to protect the 5' end of the DNA, or the beginning of the RNA. We're also going to add a 3' poly A tail, so a whole bunch of adenines tacked on to the end of the RNA. And then the last thing we're going to do is we're going to cut out all of those non-coding regions of the gene. Remember, we said that genes are arranged by introns and exons. Exons are going to be the part that are eventually going to become the protein. And the introns, they're not really doing much of anything, so they need to be removed from the RNA. And so that's the third step that's going to happen, and we call this splicing. This is where we cut out all the introns, and then we glue all the exons together. And so now we have a strand of RNA that has a 5' cap, it's got this poly A tail, and now all you have is the coding regions of the gene. So all of these exons are ultimately going to be converted into amino acids after or during translation when this takes place. And so when we're talking about transcription, you're talking about turning on a gene. If you turn on a gene, that means it's being transcribed. That means you're making a copy of RNA that correlates to that original DNA sequence. And so the next phase is translation. And so we have this nice new shiny little RNA molecule that we've made. Now we're going to use that as a template in order to make protein. And so if you look at this right here, so here's the original DNA template strand. So first thing's going to happen is going to be transcribed. And again, when you transcribe, you just pair it up with the complementary nucleotide. The only difference is where there was a T, there will be a U now, because there's no thymines in RNA. They're replaced with uracils instead. And so you're going to go through transcription. RNA polymerase is going to make your new strand of RNA. That RNA is then going to be processed in the nucleus. You're going to add the 5' cap, add the poly A tail, and then cut out the introns. And then once this RNA molecule has been processed, it's going to leave the nucleus, and it's going to go and it's going to meet up with a ribosome, because the ribosome is the organelle that's responsible for making protein, right? And so transcription and the RNA processing is occurring in the nucleus. But after that is all wrapped up, then the RNA can leave the nucleus and go and bind to one of the ribosomes on the rough endoplasmic reticulum, which is where this protein synthesis is going to be carried out. And so the way this works in your cell, we have our piece of mRNA. And so what's going to happen during translation is the ribosome is going to come and read 3 RNA nucleotides at a time. And so each set of 3 nucleotides is called a codon. And there's several different combinations of codons. Here we see AAG, this one's UCA, another one's GUC. So there's all these different combinations of nucleotides in series of 3. And so each one of those is called a codon. And each codon codes for a particular amino acid. So in other words, this AAG right here, that tells the ribosome that it needs to put in a lysine as an amino acid. Then the ribosome will scoot down, it'll look at this codon, this UCA, and that UCA tells the ribosome that a serine amino acid needs to be added next. Same thing here, you have a GUC codon. The ribosome's going to read that, and it knows that GUC is always going to match up with valine. So during transcription, your ribosome is just sliding down the RNA molecule, reading the RNA in these sets of 3s, in these codons. And then each particular codon is going to represent a various different amino acid. And so here you can see this is what is called a codon chart. And so a codon chart shows you all the different possible combinations of those 3 nucleotides, or of those 3 codons. And you can see that, well, there are 21 different amino acids that are present in humans. And so they're all kind of listed here. But each set of codons is going to be able to code for a particular amino acid. So you can see valine, for instance. Valine is always going to start with a G in the first codon nucleotide, and then that's going to be followed by a U. So all of the codons that start with GU are going to end up resulting in you putting in a valine amino acid. Just like if you look at isoleucine right here, another amino acid, these are all going to start with a U. And so there's going to be 3 of these codons that are capable of coding for one amino acid. So it's not like there are, because there's 64 possible codons, but there's not 64 possible amino acids. There's only 21. So in other words, you can have multiple codons that code for the same amino acid. So if this is the ribosomes going down the stretch of RNA and it comes across in AUA, it's going to put in isoleucine. Let's just say the next codon is AUU. Well, it's still going to put in an isoleucine. So all the ribosome is doing is reading that codon, and then based on what that codon is, it knows which amino acid to bring in and add to the protein chain. So as far as testing is concerned, I would never have you memorize this. So this will be printed on your exam, so you will have access to this. And also on your homework, because you're going to be translating, this is already on there as well. And so all you're doing is looking at the codon, finding where the codon is in the chart, and then just plug it in the proper amino acid. It sounds pretty straightforward, right? So let's do a quick little example before we get into the not so simple part. All right, so let's just go ahead, let's just make a DNA molecule. Let's go ahead and transcribe it and translate it. So, I don't know, let's just GCATCCG. Perfect. Okay. So if this is our template strand of DNA, and we're going to transcribe it into RNA, what's the RNA sequence going to be? A, yeah. Right, so you're just looking at the template strand, and then you're figuring out what the complementary nucleotide is. So T is always going to bind to A, T is always going to bind to A, G to C, so forth and so on. But in our RNA molecule, there's not going to be any thymines. Instead, those thymines are going to be replaced by a uracil. So transcribing is easy enough, right? Now let's go ahead and translate this. So this is going to be one codon, this is going to be another codon, and this one's going to be another one. So what does AAC code for? Asparagine, right? AAC. And so we know that the first amino acid in that protein is going to be asparagine. All right, what about GUA? What's GUA? Valine. Yeah, over here in the bottom left-hand corner. So the next amino acid is going to be valine. All right, what about the final one? GGC.
Glycine.
Glycine. So the next amino acid is going to be glycine. So that's translation. It's easy enough. All you got to do is transcribe it first, and then based on your RNA, just compare it to the codon chart in order to figure out which amino acid is going to be placed in that protein chain. Yes?
So do we actually write it out like that, or do we write the letter?
Yeah, so like for on the homework, for instance, and even on the exam, I would give you like a template of DNA.
No, I mean on the protein.
Yeah, so I mean you could write it, you could write out the whole thing, but there's abbreviations for a reason. And so you're not writing out these long amino acid names.
I guess I'm asking about the letter that's beside the abbreviation on the chart.
Oh yeah, well you can do it like that as well. That's fine. Actually on your homework, it wants you to only use the first letter. It doesn't want you to write arch. So that's how you're going to end up getting this phrase if you do it correctly. But as far as the test is concerned, I'll probably just, have you write it like this. But you can write it in the single letter form too. That's fine with me. It doesn't matter. Okay. One other thing that we need to mention is that, we said there's 64 different codons. 61 of them are going to code for an amino acid. And then the other 3, these are going to be kind of specialty codons. They're called stop codons. And so this stop codon is going to be important because as we go through translation and as you're building this protein, translation has to know when to stop, just like transcription did. And so once the ribosome reaches a stop codon, it knows that translation is over and that the protein's been finished. And so there are 3 different stop codons, and you need to know what all of them are. There's UGA, UAA, and UGG. And so if you look at the codon chart, you can see, so here's UGA, it says stop codon. So this stop codon, it doesn't make an amino acid. It's still a codon, but it just signals to the ribosome that translation is over. So this stop codon is going to come into play when we talk about the termination phase of translation. And just like there's a stop codon, there's also something that's called a start codon. So this tells the ribosome where to start translating. And so the universal start codon is going to be AUG. And so AUG is always going to code for methionine. Yeah?
You said there's a total of 64 and 61. 3 of them were stop codons, right?
And this one, the start codon, this one actually does. So this is included in that 61.
Oh,
The
so it's part
last
of those
one
3.
say tries.
Because methionine is an amino acid.
UGG is tryptophan.
It's on there.
I mean, not U, sorry, it's UAG. I don't know why I wrote that. Yeah, sorry, UAG. And so you have 61 that code for some sort of amino acid, which includes the start codon. It's just the stop codons that don't make any particular amino acid. And so these start and these stop codons are going to regulate when when translation begins and when it ends. And so we have our DNA, it's already been transcribed into RNA. And so this RNA molecule is going to always start with an AUG. The reason why is because every single gene starts with those 3 nucleotides. And so when you transcribe those, you're going to end up with AUG, and AUG is always going to code for methionine. So, which amino acid is going to always be the first amino acid in any protein? Methionine, right? Because the start codon is always going to be there. Every single gene is always going to start with AUG. And so, here's the beginning of the gene, and then once we get to the end of the gene, or of the RNA, we have one of these 3 stop codons present. And so the ribosome will start at the start codon, it'll start putting in these amino acids to make a protein chain, and then once it comes in contact with the stop codon, then basically the ribosomes fall off, and translation is done, and you end up with a nice little protein being made. Questions? Yeah.
Was methionine, is that the promoter?
No, methionine is one of the amino acids. So if you look at the structure, so here's my DNA molecule. This is DNA. So every single DNA molecule is going to start with this TAC and ATG. Now upstream of the DNA, this is where the promoter is. And so eventually you'll reach the end of the DNA, and then this is where that terminator sequence is in terms of transcription. But after you transcribe this, so now you have your gene that's starting with AUG. And so that AUG is going to signal to the ribosome to start making protein here. It's going to go down, come across the stop codon, which is actually going to be, the last 3 nucleotides that are associated with the end of the DNA strand. So you've got the beginning of translation, which is the start codon, and then you've got the end of translation when that ribosome comes across one of those 3 stop codons. Yeah, Alan.
If you don't get AUG at the beginning or one of those 3 at the end, you got a problem.
Exactly. Exactly. Every single gene is going to start with AUG, and every single gene is going to end with one of those 3 stop codons.
So a gene is a protein?
A gene is, so when we're talking about genes, we're talking about DNA. Because our DNA is separated into various different genes. Here's a gene, next to another gene, so forth and so on. And so during transcription, we're using that DNA or that gene as a template to make RNA. And then that RNA will be a template to make this amino acid sequence, which makes up the protein. Other questions? Yeah.
I'm confused where the AUG comes in.
Okay.
We're starting the DNA with the TAC, right?
Mm-hmm.
And then the second line of DNA is the ATG.
Uh-huh. So if this is the template strand right here, TAC, when we transcribe that, what do we get?
You're gonna get U,
I'm sorry, if it's, the ATG is the template strand. I'm sorry.
okay, so to get the RNA you're asking, right?
Yeah,
So it'll be UAC. If you start with ATG,
You put a T instead of a U.
where? Up here?
right,
Yeah, this is DNA.
okay.
So AT,
Hmm.
sorry, ATC. Let's see, methionine should be on here. It's AUG though. Doesn't make any sense.
Because
I know it's
You
AUG.
got a U.
TAC is your template.
So if you start with the TAC and you transcribe, you're gonna get AUG. Yeah, so it is ATG, see?
Huh?
Okay, so it's just which one is the template strand. So this is, the template strand is where you make the, you know, based on the, you make the RNA based on the complementary from the DNA. So if it starts with TAC, when you're making the RNA, you're gonna end up with AUG.
Okay, that's what I was forgetting was
Okay.
RNA is made from the first strand, not the second.
Right, right.
Where did that T come from?
Where did what come from?
You said the first one is the DNA strand and the second one is the template?
Well, this is, imagine this being double-stranded, right? So here's the first 3 nucleotides on the template strand, and here's the complementary nucleotides on the non-template strand. And when you're making RNA, you start with the template strand,
Oh, okay.
and then
okay.
put in the complementary RNA nucleotides to build your RNA. Okay, makes sense? Are you being sarcastic? Very sarcastic. Okay, I feel like I've lost some of y'all, so let's back up a little bit. Okay, let's make some room.